I am Kimberly Luddy, and we are at AACR, presenting my poster with work that was done at Moffitt and Trinity College Dublin. The work that we did is looking at tackling radiotherapy resistance in advanced prostate cancer. So the idea is that radiotherapy traditionally works when a patient comes into the clinic, but then over time the radiation stops to be effective and the patient develops resistance to radiotherapy. So we want to come up with new and novel ways to attack the cells that are resistant to the radiation. And so it has been shown that DNA damage induced by radiation will actually trigger the expression of NK ligands on the surface of surviving tumor cells. And these NK ligands can actually activate NK cells. NK cells are immune cells within the body that can kill target cells. And so if the NK cells become activated, they can then kill the tumor cell. So our hypothesis is that radiation-resistant cells are likely more sensitive to NK cell-mediated killing. And so we used an isogenic cell line model developed to study the difference between a radiation-resistant cell line, which we have in red, and a radiation-sensitive cell line, which is represented in green. And we see that when we look at these two cell lines and we look at the surface of the receptors of the ligands on the surface of the tumor cell lines, we actually see that the radiation-resistant cell line has increased NK ligand, meaning that it's likely that they induce NK cell activation. And we also look at their sensitivity to NK cells. So you can see our radiation tumor cell line is more sensitive to NK cell-mediated killing, showing that our hypothesis was correct, that radiation resistance could be targeted by NK cells. Interestingly, when we combine the two cells together in a single well and we compete them against each other, so we now have the radiation-resistant and the radiation-sensitive group in one well being targeted by NK cells, we see that the NK cell sensitivity remains. Therefore, if you have a heterogeneous tumor with different radiation resistance levels, you can still get direct targeting of the radiation-sensitive population. And we see that when you have different profiles of radiation resistance, so you have high levels of radiation sensitivity, low levels of radiation sensitivity, and you challenge them with NK cells, you actually see that the very radiation-resistant patient would benefit even more from NK cells than, say, a radiation-sensitive patient. Therefore, the addition of NK cells to patients who are no longer responding to radiation therapy could be beneficial. And we again look at this with combining the two therapies. So this is where we take radiation and then we give NK cells. And we can see that the two therapies are better than the individual therapy and that you have more tumor cell killing when you combine the treatments. And this is when you look at the populations in separate cultures and when you look at them in cultures together. You can see that over time, the NK cell combination with radiotherapy is highly effective. And using a mathematical model, which you can see back over here in our methods section, we use a Lavoterra style model to then take our data a bit further. These assays can only go out for about a week. So the in silico modeling can actually tell us what might happen if we were able to continue them on longer. So this goes out to 3 weeks. And you can see that if you give the two treatments together, you actually get an extinction event. So this is representing NK and RT treatment. And what we mean by extinction is that both populations of cells, the radiation sensitive and the radiation resistant population, both go extinct and the tumor is cured.